All right, if you're watching this video, there's about 15 minutes before lecture actually starts. So your first word for attendance, and don't forget to take a screenshot every time someone says an attendance word. Anyway, your first word for attendance is Sanyo, like you can see on the screen, S-A-N-Y-O. Good morning.
I couldn't find the lecture with, where we talked about the.
Yeah, did you see, I emailed you back.
I
So
didn't see it.
the last announcement that I sent had a link to it.
Oh, did it?
So if you go to the announcements, well, there's two. First of all, the last announcement has a link to the announcement. There's an announcement that says something like photosynthesis lab. Everything you need is there. The data's there, the links are there, the video is there, it's all there.
Awesome. And that's what, due tonight at midnight, I'm guessing?
Yeah.
Okay, awesome. All right, thank you.
Morning.
Good morning.
Did you have a good spring break?
Yes, sir. I was decided to stay here. This is great.
You stayed here?
Yes, sir.
That could be good too. You do anything or just kind of relax?
I just kind of relaxed, kicked it. Nothing much for real.
I did that too. I was planning on getting more work done,
Mm-hmm.
but man, it was nice to just,
Yeah,
To do nothing for a bit.
Yeah.
I got a little work done, but not nearly what I was planning on. How about you? How was your spring break?
Uneventful.
Uneventful? Good. No news is good news. Right. Did you go anywhere or did you stay here?
I just sit here. Well, the weather was good, so.
Yeah.
You can't complain.
It was. Excuse me. All right, we've got less than a minute left. We'll get slowly started. As a reminder, if you're online, you can send in the attendance words sometime before 9 a.m. If you're watching the video, so none of you, if you're watching the video at a later date, you need to take a screenshot every time I say one of the attendance words. And the last attendance word, if you're going to call it that, is going to be me asking you to ask me a question as usual. So basically everything's happening as it usually does. I'm just giving you a reminder since we've had a week off. So just reminding you how things go. Yeah, that's really it. Today, once we get started, we are going to finish up chapter 7 and jump into chapter 8. And it is 8 o'clock, so that's what we will do right now. So again, if you're online, don't forget you have to send those attendance words. Just because you're logged in doesn't mean you are counted as present. You have to send in the attendance words. All right, let's jump to it here. Bear with me while I get this pulled up, please. Here we go. All right. All right, so this is where we left off talking about. The last discussion we had on chapter 7, chapter 7 was all about meiosis, right? So the last discussion we had was what happens when meiosis goes wrong. And this is what we left off with. We talked about one of those examples, which is trisomy 21. You should be familiar with this because you slightly familiar. If you've done the lab already, which you should have because it's past due, you looked up trisomy 21. But we put a big old X to this, indicating I'm almost definitely not going to ask you any questions about trisomy 21. But anyway, we'll talk about it because your textbook talks about it. This is when you have 3 of the number 21 chromosome. Again, you're supposed to have a pair, right? You're supposed to have a pair of each chromosome, not 3, not 1, not 4, but 2. So in this case, you have an extra. This results in 47 chromosomes total instead of 46. And a person who has trisomy 21 has something called Down syndrome. I'm sure you've heard of it. You're probably familiar with it. This affects about 1 in every 850 children. It is the most common chromosome number abnormality, and it's the most common serious birth defect in the United States, which brings up a good independent work topic if you want to look it up. First of all, what's the second most common serious birth defect in the United States? Second of all, what's the most common serious birth defect in the entire world? Or pick a country, you can look that up. The risk does increase with the age of the mother. So women who are 35, pregnant women who are 35 years and older are candidates for prenatal screening. So again, another independent work topic. Why? What is it about getting older for women that makes them at higher risk? Especially when you think about this, this doesn't happen in the womb, right? This isn't an issue of having an old uterus, so to speak. This is an issue of meiosis, right? So these are women who are created eggs. And mind you, and you can look this up for independent work too, they create their eggs when they're very young. So they just have these eggs, they're already created, and then they just use them, throughout their life. So you can look that up. Like, what is the cause of all this? I'm not going to ask you any of this on the exam, but again, that's interesting topics if you want to look into it. So the first word for attendance is this. Will I ask you any of this on the exam? So the first word for attendance is either yes or no. And again, if you're in person, you don't have to send me the attendance words, but you can if you want extra credit. Any questions about this slide before we go on and talk a little bit more about trisomy 21? All right. So people with Down syndrome have characteristic facial features. I'm sure you've seen it. You can generally recognize it. They generally have a shorter lifespan. They have varying degrees of developmental delays, and some may live to be middle age or beyond, and many of them are socially adept. So this is all from your textbook. This is why I'm including it. But again, I'm not going to test you on this. And it's also, again, in my opinion, you can look into this for independent work, right? So what is it about that extra chromosome that makes them have those characteristic facial features? What is it about that extra chromosome that often results in the shorter lifespan? What is it about that extra chromosome that results in the varying degrees of developmental delays? Just things to look into. Anyway, here's what it looks like on a karyotype, which again, you should be familiar with. You can see here, there's an extra chromosome 20. Well, no, I'm sorry. That's a normal karyotype. As you can see here, chromosome 21, there's only a pair, as it should be. And then here is the karyotype that has 3 chromosome 21s. And again, this is great topics for independent work if you want to look into it. I have a hypothesis that I haven't looked up yet, but I think one of the reasons, and I'm sure somebody knows this, but I would assume the reason this is one of the more common birth defects is I would assume a lot of the other ones are more fatal. Now, granted, you've done the lab, so you know, for example, you can have an extra chromosome 13 or an extra chromosome 18, for example. But anyway, my hypothesis is the reason this is the most common is because it's probably not fatal. And the reason I think it's probably not fatal is because look how small that chromosome is, right? Chromosome 21 is a very tiny chromosome. So if you have problems with that chromosome, it makes sense that it would be less fatal, for example, if you had an extra chromosome number 1. It's a huge chromosome. That's a lot of genes. And again, independent work out because I really don't know it. What do you call it when you have an extra chromosome number 1? Or is that even survivable? Are there any human beings that have ever had an extra chromosome number 1, or do they just, die? Does the zygote not make it? I don't know. Anyway, any questions about that? All right, really quickly, an overview. This is great timing since we've had a whole week before we talk about all this. Meiosis is the basis of sexual reproduction. When you think of homologous chromosomes, especially on the exam, you should think of meiosis, right? When it comes to questions about meiosis or mitosis, if it's about homologous chromosomes, it's only going to be about meiosis, not mitosis. Specifically, if you remember, only meiosis 1 deals with homologous chromosomes. Meiosis 2, just like mitosis, deals with system chromatids. Meiosis is the thing that gives us the gametes, which is our sperm and egg, and the life cycle of a sexual organism. Oh, excuse me, I'm reading the overview. Yeah, sorry. Really quickly, let's talk about abnormal numbers of sex chromosomes. That was a good review, but it was out of order. Anyway, so again, we just got finished talking about trisomy 21, which is where you have an extra chromosome number 21. Now let's talk about what happens when you have an abnormal number of sex chromosomes. Non-disjunction can lead to this, right? So you can have extra X chromosomes, extra Y chromosomes, or maybe missing chromosomes. It upsets the genetic balance less than the autosomes. You can look that up for independent work. Why? Why is this less important than autosomes? You've already heard my hypothesis regarding trisomy 21. It applies here too because those sex chromosomes are very tiny, but you can look it up for yourself. And the next slide, I'm going to show you list some common sex chromosome abnormalities. Here we go. XXY, which is one that you didn't have on the in the lab, that's called Klinefelter syndrome. You know what? I'm not going to ask you any of this for the exam, so you can look it over if you want. But I'm not going to ask you what the names of any of these are or how frequent people get this in the population. But it's worth looking into. This one's an interesting one to me if you want to look into this for independent work. XYY. I read somewhere they call them super males because it's the Y chromosome that makes a male a male. And I read somewhere, and you can see here the frequency is 1 in every 2,000 people has the XYY condition. But I read somewhere that in prison population, it's more frequent than that. Like the percentages are higher. And I think they reckon that because they have that extra Y chromosome, they also have extra testosterone. That makes them extra aggressive, and therefore they're probably more likely to be violent offenders, and therefore more likely to be in prison. So things you can look into for independent work if you want. But again, I'll put it next to it to make it official. I'm not going to ask you any questions about this on the exam. It is worth noting, especially in today's political climate, that sex is an interesting thing. It is sometimes not as simple as X and Y or X and X. It can get muddied. And it gets muddier than this too, but this is a 100 level class, so we don't really dig into it. You can if you want for independent work. Anyway, any questions about this? All right, so we're done talking about like extra chromosomes or missing chromosomes. But what we haven't talked about is damaged chromosomes. So sometimes there's partial duplications and deletions. So in other words, when you go through S phase of interphase and you're duplicating those chromosomes, sometimes there's a mistake there, right? Sometimes this is survivable, but usually it results, if it's survivable, results in physical or mental abnormalities, as you might imagine. If you have extra parts of the chromosome or missing parts of the chromosome, that's not good, right? Then we have these things called inversions. This is when you have a detachment of a part of the chromosome and a 180-degree rotation and a reinsertion of that part of the chromosome. And these are actually mild effects, which should make sense because you still have all the genes, all the genetic information's there, it just happens to get flipped. Of course, you would imagine at that point where it got flipped, there might be some issues, hence the mild effects. And then we have something called translocations. This is where you get a part of a chromosome that disassociates and reattaches to a different non-homologous chromosome, meaning that you might have a chunk of chromosome 1 break off and, I don't know, attached to a chunk of chromosome number 5, for example, right? This can be benign or it could have devastating effects. Again, this is a 100 level class. I wish we could talk more about it. But a lot of evolution actually came from this. This is where we get a lot of differentiation between species. But again, we don't have time to talk about that. You can look into it if you want. So basically what I'm saying is when you have big effects on chromosomes, that often, assuming it's survivable, will lead to different, could lead to different species. Obviously not always. Anyway, here's some pictures of what we're talking about. Here's an inversion. Right, so we got this chunk of the chromosome was taken out, flipped around, and put right back in. Here we have a reciprocal translocation. So we have a chunk of this chromosome was swapped with a chunk of that chromosome. In a sense, not in a sense, but I don't know, when you're trying to think about it in your brain, that translocation is a little bit like crossing over in prophase 1. A little bit, only in that you have chunks of chromosomes swapping. Now, granted, in crossing over, what you're looking at is the same homologous chromosome, your mom version, your dad version, those swapping chunks. When it's a reciprocal translocation, these are different chromosomes. So again, hypothetically, I'm just throwing out a number. This might be chromosome number 1. This might be chromosome number 5. Any questions about that? All right, so I left this last part in here from the old textbook. I'm not going to ask you any questions about it, but it's once again something you can look into for independent work. Like, what are the advantages? What are the evolutionary advantages of sexual reproduction? And you can see some of them here, or in this case, asexual reproduction. What are the advantages of that? It eliminates the energy needed to find a partner and procreate with them, right? So that's an advantage. And some disadvantage, though, is that it reduces genetic variation. So again, this might be advantageous if you have a species that's sparsely distributed, right? So it's hard to find a mate because there's just not another animal around, or I guess this has to be an animal organism. Then asexual reproduction would be good. Also, if that organism is superbly suited to a stable environment, meaning that's a good fit for the environment, and the environment's not changing, then who needs sexual reproduction? Who needs to have this genetic variation? If you're perfectly adapted to your environment, the environment's not changing, it would be better just to clone yourself. And that's exactly what asexual reproduction is. Another thing this doesn't even mention, when you think about it, there's a lot of insects and arachnids where it's deadly, right? Reproduction is deadly. The male often gets killed after having sex with the female. So, hey, there's an advantage, not getting killed. Anyway, you can look into that for independent work. Again, I'm not going to ask any questions about it. Really quickly, this is regarding sexual reproduction as opposed to asexual. You have advantages because you produce offspring that have very genetic makeup. So that would be good, And again, you can get into it for independent work if you want. We're going to talk more about that bullet point when we talk about evolution. Anyway, look it up if you want. It is now, or again, as a reminder, I'm not going to ask you any questions about this on the exam, but since it's on the screen, the next attendance word is, let's see, evolutionary. So one of the evolutionary advantages of sexual reproduction is that it reduces the incidence of harmful genes blank. More what? So in this case, the attendance word is on the screen. Reducing incidence of harmful genes more blank. There's one word, it's on the screen. That's your next attendance word. All right, so what time is it? 8:14. So now, hopefully everybody who's going to be here is here. Let's do announcements and reminders. First of all, we're past the midterm. I'm going to submit the grades. They're due by Wednesday. I hope you get them submitted today. I have to submit grades and attendance. And also, after I do that, as a reminder, I'm going to input your independent work grades into Moodle. Moodle's not official in WVSU Online. It's not official, but once I put it in there, again, unless you've done all your independent work, you're going to see your grade drop. And the reason I'm doing that is because I want you to know what your actual grade is now. I don't want to do that at the end of the semester where you say, oh man, I've got a C, I'm doing great. And then I end up putting in your independent work grade and it drops to a D, right? I don't want you to be surprised. Also, I forgot the exact date, but this is the last day, sometime this month is the last day to drop this course. I hate to be negative, but some of you, not you guys, because I'm literally preaching to the choir, but the people who are probably not here or online or probably not even watching this video, you should probably consider dropping this course. Historically, I've seen a few people have an F at midterms and bring it up, but very rarely. That being said, if you are one of those people with an F at midterms and you do want to pull it around, I'm here for you. It's going to take a lot of work, and I'm here to help you. We'll get you caught up on labs, get you caught up on independent work. I'm here to help you, but if you're not here to work, I highly recommend dropping the course before it's too late. Any questions about midterms? All right. So as a reminder, exam 2 is past due. That was due Sunday. And it's also now too late to retake exam 1, which also means, also as a reminder, if you retook exam 1, your grade didn't change. You have to tell me that you retook exam 1, and I have to go in there and manually average your first grade and your last grade and change your grade. So if you retook it, please let me know. As far as I know, no one did. And going back to grades, sorry to sound harsh, but I know there's going to be at least 2 or 3 people at the end of the semester like, is there anything I can do about my grade? And I'm going to remind you of this discussion. I'm like, you didn't even retake the exam. You had the chance to retake the exam and you never did. But anyway, now that it is too late to retake exam 1, you can now meet me online for feedback. I will go over that exam with you and give you all the correct answers. And we'll talk about your incorrect answers and talk about why they were incorrect. And maybe I can talk to you and help you remember the correct answer, give you some hints, like a tutor would. And not only will you get all the correct answers, which you're going to need because that's what you need to study for the final exam, but I'll give you a 5% boost to your exam grade. So the offer's there. Please meet me online. Let me give you feedback on exam 1. Get your 5% boost. Get all the correct answers. Any questions about exam 2 and exam 1? Actually, one more thing. Sorry. I'm just going to go ahead and say I haven't even looked at the results yet. But exam 2, I'm going to let you retake it. I don't even know what the results are. But just like exam 1, sometime in the next few days, I'm going to go in there and change the settings. I'm going to let you retake exam 2, just like I let you retake exam 1. So just like exam 1, you go in there, you retake it as many times as you want. The retake is open book. It's not timed. And what you'll do is you'll tell me what you're satisfied with your new grade. You'll email me, and I'll average your first grade and your highest grade. And again, just like the retake for exam 1, if for some reason you keep getting a lower score on this retake that's open book and closed, not timed, then we'll just keep your original higher score. There's nothing to lose. That being said, now are there any questions about exam 2 or exam 1? All right, let's quickly talk about labs. I had someone asked me this on email, I forgot who, but it was a great question. After this week's lab, all the labs we're going to be doing are virtual. They're all, online. They're using computers, which makes sense because we're going to be talking about things like evolution and stuff like that. You can't really do evolution in a laboratory, but you can simulate it online. So yes, they're all virtual after this week, but you still have to be present, right? There are some exceptions, and you'll know about them when they happen. So for example, if you do next week's lab, I think it's on DNA, if you go ahead and do next week's lab, submit it before next week, then most likely I'll grade it and say, hey, great job. You don't have to come into the lab. So I'm not saying you don't have to come into lab, but if you do it early, I might tell you specifically that you don't have to come into lab. So keep that in mind. That's just some incentive for you to go ahead and do the labs if you want. And then there's also a reminder, the lab report is due today. So as a reminder, it was supposed to be due 2 weeks after the lab itself. But 2 weeks after the lab itself was this past Wednesday, and that was spring break. So I didn't want to do that. So today is the due date for the lab report. Remember, it's worth 20 points, so it's worth basically the entire lab. And if you have any questions about it, obviously come to me, I can help you. But also, if you go to the announcements, there's a section that says, I think, like photosynthesis/respiration lab. Everything you need is there. The data's there. The video of us talking about how to do a lab report's there. The file, the document that you need to do the lab report is there. The link to where you need to submit that document is there. Everything you need is there on the announcements. But again, I'm here to help you. The lab report is not a test. So if you need help, come to me. I will help you write it. I'm here to help you. And then finally, the last announcement reminder, this is slightly different than what I've told you before. I've added a date. So the Friday before and the Monday after Easter, we're not going to meet in person. It's going to be 100% virtual. So anyway, that's it for announcements and reminders. Any questions about that? All right. So I guess the next word for attendance is going to be more like a number. Lecture and office hours will be 100% online on April 18th. And what other date? All right. So no more questions about that? Or no questions at all? All right, that's the end of chapter 7. Let's quickly jump into the next one. We're a little bit ahead of the game, which is a good thing. But those last chapters that we cover, they're really, I don't know how to put it, long, I guess you could say. So it's kind of good that we're ahead of the game. All right, where are we at here? Downloads. Here we go, chapter 8. Chapter 7. I don't remember. My brain is so used to the old textbook because I taught with that for like 9 years. I want to say 21, but this new textbook is a different number, so I'm not quite sure. Also, to your point, or maybe not to your point, but I'll remind you, the last chapter we talked about, and also chapter 6, that's all stuff that's going to be on exam 3. So we're still covering stuff that's going to be on exam 3. And this is about patterns of inheritance, i.e. genetics. So we're finally about to get into genetics. In my opinion, this is one of the more important chapters as far as you guys are concerned. You're non-biology majors, right? So this chapter is very relevant to your everyday life, to understand how you work, to understand how viruses work, and some vaccines, right? This is very important information. Here we go, let's get into it. So this is from the old textbook. Again, I keep some of the stuff from the old textbook. At the beginning of the chapter, they always have why does this chapter matter? And of course, you can write a paper about that if you want for independent work. Why does genetics matter? But let's talk about some of these pictures. So, for example, how would you describe that woman's hair color? What color do you think that is? Red, that's how I would describe it too. But I'm just guessing, and I don't know this for sure, but that's probably not real, right? That's probably fake hair color. She probably dyed it like that. And then, of course, you can see this woman has some tattoos. Now, I guarantee you, when this woman has kids, those kids are not going to come out with that hair color or with tattoos, right? Because not everything is inherited, right? Some things are not inherited. And that's part of what we're going to talk about in this chapter, right? So just because something happens to you or your body does not mean you pass that along in your genes. Some things you do, some things you don't. This is a picture, obviously, of a man and a child. That happens to be a daughter. The old textbook, what they were getting at is it's the man that determines the sex of the baby. Because a female, as you learned from the last chapter of meiosis, a female is an XX, right? So when a female goes through meiosis and those X chromosomes segregate into different cells, it doesn't matter. 100% of her eggs are going to have an X chromosome. Men, on the other hand, are XY. Therefore, when they go through meiosis and those segregate, that means 50%, we're all men in this room right now, 50%, if you were to look at your sperm, 50% of them, about 50% of them would be X chromosomes and about 50% of them would be Y chromosomes. So it is the man, therefore, that determines the sex of the baby. I don't even remember what that picture was getting at. I think it has something to do, all right, hold on. I wrote it down. Your chances of inheriting and passing along a genetic disease can often be determined from your family history. So that's what that picture is about. It's talking about family history. And yes, that is one of the things that's very relevant in this chapter to your everyday life. We talk about genetic diseases. So even if there's no genetic diseases in your family, the person that you mate with might have genetic diseases in their family. And this is some information that'll help you understand that. Anyway, let's jump into it. This is another thing from the old textbook. They always start off with a little story. I'm not going to go through it. You can download the PowerPoint and read it if you want. I'm not going to ask you any questions about it. There we go. So once again, I'm using the old textbook. This PowerPoint is based on the old textbook. I think the old textbook did a better job than the new textbook on this chapter, as far as this chapter is concerned. I like the fact that they kind of tell it as a story. So instead of me just giving you information, they kind of tell a story, and I like to stick with that. And the same thing will happen when we talk about Darwin, for example. But anyway, the only reason I bring that up is because if you're the kind of person who likes to follow along in the textbook while I teach, just so you know, it will be a little out of order, and you'll see some pictures and some things that are slightly different. So let's jump into it. Let's talk about genetics and heredity. First thing you need to know, and I might ask you this on the exam, is what is heredity? It is the transmission of traits from one generation to the next. That is the definition of heredity. Now, even if I don't ask you what the definition on the exam, which again, I don't remember if it's on there or not, you definitely need to know what it means, right? Because it's a word that we're going to use a lot. All right, so the next word you need to know, because I might ask, but definitely because we're going to use it, is genetics. Genetics is the scientific study of heredity. So you know what heredity is, now you know what genetics is. It's the scientific study of heredity. So let's talk about Gregor Mendel, who's that picture at the top right. I heard there was a noise. If somebody has a question or anything that's online, go ahead and unmute yourself and ask it. All right, if not, let's talk about Gregor Mendel. In the 1860s, Gregor Mendel argued that parents passed their unique genes to offsprings. He was the first person, at least in history that was recorded, to suggest this. Of course, he didn't call them genes. Back then, he just called them heritable traits. We didn't know what genes were. Actually, let me rephrase, let me back up, let me take a step back. He was probably not the first person to say that part, right? That part is not unique, right? Parents pass the unique heritable traits to their offspring. I'm sure everybody knew that, right? Like, you get two tall people, they have kids, probably gonna have tall kids. Two short people have kids, probably gonna have short kids, right? People knew that. But that's where we start. It was these genes, or again, as he called them, heritable traits, not that I'm gonna ask you that, but it's the genes that were responsible for these inherited traits. So that's where it started getting new. So again, people knew, like, parents passed down their traits, but he started thinking there's something in you biologically, right? Something in your cells that is passed along, and that's what's responsible for these inherited traits. He also argued that these genes, and again, he didn't call them genes, we didn't know what genes were, he called them heritable traits. He argued that they retain their individual identities from generation to generation, no matter how they're mixed up or temporarily mixed. So that was a new idea as well. So for example, and I'll just use myself as an example, I have blue eyes. My mom has brown eyes, my dad has brown eyes. So what happened there, my mom has one gene, now technically an allele, but we'll talk about that later. She had a gene for blue eyes and a gene for brown eyes. But the gene for brown eyes wins, if you want to call it that, right? So because of that, she had brown eyes. My dad had a gene for blue eyes and a gene for brown eyes. but because the brown eye gene was more dominant, he had brown eyes. But they both carried that gene for blue eyes. It was temporarily masked. So then once they had me, I had blue eyes, and then the trait came back. And we're going to explain how that happens in this chapter. But yeah, again, at the time, that was a new idea. So any questions about this slide? I will say, other than the first 2 bullet points, there's nothing important on this slide as far as the exam is concerned. The rest of this is basically just an introduction to what we're about to talk about. So, that being said, let's talk about it. Let's talk about what he did in Abbey Garden, which is where he did his research. Mendel studied garden peas. So that's what he did his work on. A lot of genetics, people who do genetics right now use like fruit flies very often, or a plant called Arabidopsis. Not that you need to know any of that. But he used garden peas. Why? First of all, they were easy to grow, and also because they come in readily distinguishable varieties. So easy to grow, that makes sense, right? If you want to study heredity, you probably don't want to grow redwood trees, right? Because it's going to take them hundreds of years to get big and mature and be able to reproduce, right? So you don't want that, you want something easy to grow. Also, the readily distinguishable varieties. That's important. Like, so say, for example, if you wanted to study, I don't know, Bermuda grass, that would be hard, right? Because they're not, I mean, it's just grass, right? So what are the different varieties? But anyway, that's why he chose peas. I'm not going to ask you any questions about that. Again, this is kind of like an introduction. So some 2 terms you definitely need to know and know the difference between. 1, because I might ask you, and 2, because we're going to use them a lot. Character versus trait. Character is a heritable feature that varies among individuals. And a trait is a variant of a character. So for example, eye color is a character. Blue eyes is a trait. Brown eyes is a trait. Green eyes, it's a trait. No, let's think of another example. I don't know. Chin dimple. That's not a good example. Anyway, yeah, so character, again, that's the feature, and then the trait is the variant, right? So make sure you understand that. We're going to deal with that a lot in the lab this week. So each character that Mendel studied occurred in 2 distinct traits, which makes it very easy to study. So for example, all right, one bad thing to study would be height, right? If height was the character, think about all of the different traits. It's not like all humans are either short, tall, or medium, but actually that's not even, it's not like they're either just 5'5" or 6'5", right? There's all these different traits when it comes to height. So that would be a bad one to study. But Mendel, when he studied his peas, everything he looked at only had 2 different traits. So, well, we'll get there. An important advantage of pea plants was that he could also strictly control the reproduction, which is obviously very important if you're trying to study heredity and genetics. It's important to be able to say, all right, I want this thing to mate with this thing, and then I can study the offsprings, and I can get these offspring and mate this offspring with that offspring, right? So he could be in control. Again, this is from the old textbook. I'm not even going to talk about it. I'm not going to ask you about it. It's there if you want to see it. That's what a pea flower looks like. You can see here you have the stamen. Here you have the carpel and the different stamens. So he could use tools that you see on this next slide, and again control the reproduction. So this flower was crossed with that flower, so on and so forth. I'm not going to ask you any questions about this. Again, this is from the old textbook. I guess the point your textbook was trying to make is that he controlled the reproduction. So nothing that happened in his experiments was by accident, right? This is all on purpose, and he knew the lineage. That's all your textbook was trying to get to. Here's some other words you need to know. When he did this, he created purebred varieties of these plants. You need to know what a purebred is. A purebred is a variety of plant that self-fertilizes to produce offspring that are identical to the parent. I don't remember if this is in the new textbook, so if you're taking notes, you might want to write that down. You might want to write that down because it might not be in your textbook, and I am going to ask you that on the exam. You need to know what a purebred is. Now, once he had these purebreds, and it took him a while to get there. But once he had the purebreds, he would cross them. Right? So, for example, a purebred flower with purebred plant with purple flowers, he would cross that with a purebred plant that had white flowers, for example. And that is what we call a hybrid. And you need to know that too. When he did that, what we call that is it's called a genetic cross. So when you cross fertilize something, that's called a genetic cross. The plants that you use to do that cross, that's called the P generation, the parental plants, if you will. And then their offspring is called the F1 generation. So if you were to relate that to humans, you could say that your parents are the P generation and you are the F1 generation. And then if you were to have children, they would be the F2 generation. Because the F2 generation is a cross of the F1 plants. I also want to point out that this is, again, a 100 level course. It's a very simplified version of how things work. So now when we do genetic crosses, like people who do genetics on plants, it gets a lot more complicated than just P, F1, and F2. And you can look that up for independent work if you want to. So the next word for attendance is, if you were to cross the F1 generation, what would their offspring be called? Cross of the F1 plants is known as the blank generation. So I guess it's really not an attendance word, more like a letter and a number as a hint. All right, any questions about this? You guys got to bear with me. I admit that this part was very boring. It gets better. It's like all the background information that you need to understand before we can get into the good stuff. Speaking of which, let's talk about Mendel's law of segregation. You're already familiar with the law of segregation because you learned about meiosis. But now we're going to look at it from a different context. So Mendel performed experiments in which he tracked the inheritance of characters, for example, flower color, that's the example I used earlier, that occur as two alternative traits. So like I mentioned earlier, there's either purple flowers or white flowers. There's nothing else, at least with these pea plants, right? So there's no purplish white flowers, or there's no purple flowers with white spots or anything like that. There's two alternative traits. And the results of these experiments that he did led him to formulate several hypotheses about inheritance, and you need to know about them. I'm going to show you his hypotheses after this next slide. So again, not that I'm going to ask you any questions about this, but the things he studied only had two traits, right? So a flower was either purple or it was white. It was either axial, meaning it was somewhere on the side of the plant, or it's terminal, meaning it was at the end of the plant. The seeds, right, the peas themselves were either yellow, should be a W there, but it migrated down there. It could be yellow or round, right? Excuse me, yellow or green. They were either round or wrinkled. The pea pod itself was either inflated or constricted, right? So he had all these things he was studying that had two distinct traits. Not an in-between thing, but only two. So let's talk about, what we're getting to is hypotheses, but first we need to talk about monohybrid crosses. A monohybrid cross is a cross between a purebred plant that differs in only one character. So for example, again, when he's doing these studies, he's like, all right, let me cross this plant that has purple flowers with this plant that has white flowers. That's what you call a monohybrid cross. So you already know what a hybrid is. You already know what the prefix mono means. So that should make sense, right? So you're looking at a hybrid, you're making a hybrid when you're looking at just one character. But here's where it got interesting. I mean, you might not find any interesting. He did. When he crossed these purebred purple flowers with purebred white flowers, 100% of the time, the flowers were purple, like the offspring were purple. So that was interesting, right? To him, he's like, well, what happened, right? So why is it that every time I cross a purebred purple flower plant with a purebred white flower plant, why is it that 100% of the time, and he did it hundreds, probably thousands of times, 100% of the time they're always purple? And he didn't understand why. Here's a visualization of what I'm talking about. So again, crossing the purebred purple flowers, purebred white flowers, 100% of the time, they were all purple. What I don't like about this picture is it kind of looks like he did this once. Like, all right, I made it this one plant with that one plant, and I got this one plant as the offspring. That's not the case, right? He did it many times, and 100% of the time, the F1 generation all had purple flowers. Then, where it gets even trickier, he would cross the F1 generation, right? So again, I don't like this picture because when these two mated, it's not like they just reproduced and created one plant, right? When these two mated, there were a lot of plants, which this picture doesn't show. But when he crossed that F1 generation, those like two purple flowers that were in the F1 generation, and he did it again hundreds, maybe thousands of times, this is the ratio he got. So about 75% of the offspring had purple flowers, and about 25% were white. And that was the, that was the ratio. That's a ratio you need to know. I'll talk about it again later. But that 3-to-1 ratio is something you're going to need to be familiar with. But yeah, that's how it worked. Thousands of times he did this, and that was always the results. About 75% were purple, and about 25% were white. So that's why he said, like I said earlier, he knew that there was these genes, he didn't call them genes, but he knew there was something in there that was temporarily masked, right? So he saw that 100% of the F1 generation was purple, but he knew that there was something in there that still had the instructions how to make a white flower. So when he crossed them, again, 25% of the time, that white flower did show back up. So the white flower gene did not disappear. It was just hidden when the purple gene was present. And again, he didn't use the word gene, but that doesn't matter. As a matter of fact, me using this word gene is actually incorrect. You'll learn why later. There's a better word for it. But for now, we'll just say gene to keep it simple. So he figured out that those F1 plants carry two genes, one for purple and one for white, right? So he knew they had to be there. Otherwise, that F2 generation wouldn't have any white flowers. So obviously the F1 generation did still carry that gene for the white flower. And obviously too, he figured out that the purple flower gene was dominant to the white one, right? Whenever the purple flower gene was present, it won, so to speak, right? You would see that trait instead. So from these results and some other stuff, some other experiments that he did, he formed 4 hypotheses, and you need to know about them. The first one is this: there are alternative versions of genes that account for variations of inherited characters. You need to know that. I mentioned that before. The first time we talked about homologous chromosomes, I mentioned that. I was using some hypothetical examples, like, for example, if there's a gene for back hair on the tip of chromosome number 7, that means for every human at the tip of chromosome number 7, there's going to be that gene for back hair. There might be different versions of it, like you don't have back hair or you do have back hair. But yeah, those are alternative versions. And going back to the example I used before, the eye color, right? So there's a gene that controls eye color. There's different versions of them. I only have the blue version. My mom had a brown version and a blue version. My dad had a brown version and a blue version. There's alternative versions. There's going to be a name for that. I'm going to give it to you soon. Actually, you already know it if you've done pre-lab. So here it is. Alleles. So now finally I can start using the correct terminology now that I've given you this word. So when I was talking about the gene for a purple flower and the gene for the white flower, that was incorrect. The proper thing to say was allele. So there's one gene responsible for flower color, but there's 2 different versions of them, the purple version and the white version. These versions are called alleles. Any questions about that so far? All right. So the second hypothesis: for each inherited character, an organism inherits 2 alleles, one from each parent. I'll say this: you do need to know that for the exam, but I'm going to go ahead and tell you now. Later in the chapter, I'm going to come back and say this isn't exactly 100% correct. For the sake of teaching genetics, we keep things simple, and then we make them a little bit more complicated. So I'm going to go ahead and tell you now, you do need to know that, but there are exceptions to this rule. I mean, think about that, right? Again, hair color would be a great example. It's not like everybody either has black hair or red hair, right? There's like different shades of black, different shades of red, different shades of blonde, right? So that's a great example. So it's not, even though you need to know this, I will tell you there are exceptions to this rule. And also, again, these are just his hypotheses, right? These are his 4 hypotheses. Any questions so far? All right, so again, just to keep things simple, if we're thinking about a trait that has 2 alleles, then here's some terms you need to know, and again, you should already know them because you did the pre-lab. An organism that has 2 identical alleles is called homozygous. So for example, I'll just again use myself as an example. I have blue eyes. That means I have 2 recessive alleles, and I'll talk about recessive later. But therefore, I am homozygous when it comes to that gene, because I have 2 identical alleles. My parents were heterozygous, because they had 2 different alleles. So again, my mom had the brown allele and the blue allele. My dad had the brown allele and the blue allele. So they are heterozygous. These are words you need to know. We're definitely going to use them a lot in this chapter, and we're going to use them in lab. So homozygous, you have 2 of the same alleles. Heterozygous, you have 2 different alleles. And this shouldn't be that hard for you to remember, because I'm sure you're already familiar with the prefixes homo and hetero, right? Homo, same, hetero, different. And thanks to the last chapter, you are also familiar with the term zygote, right? So just think about that, and hopefully that'll help you remember. Any questions about the number 2 hypothesis? All right, the next word for attendance. If an organism has 2 identical alleles, how would you describe them? What one word would you use to describe an organism that has 2 identical alleles? Such as myself, with my 2 identical recessive alleles for my eye color. All right, if there's no questions, we'll move forward. Here's a meme that I found. Again, I don't make these memes up, I just find them. And I'm also a dad, so it's like dad jokes. I like stupid humor like this. Anyway, number 3. If 2 alleles are different, so if you're heterozygous, one of those alleles is going to determine the appearance, and it is called dominant. And the other allele has no noticeable effect, and it is called recessive. These are definitely words you're going to need to be familiar with. We're going to use them a lot in lab. Dominant, recessive. When we write them, as you should in lab, we use an upper letter to represent dominant and a lower letter to represent recessive. Which is a good time for me to remind you, when you fill out the tables, there's 2 tables you need to fill out for the pre-lab. Like say, for example, you have a chin dimple. That means you have the dominant trait, meaning you could be homozygous dominant, which is where you have 2 dominant alleles, or you could be heterozygous, which would be written like this: 1 dominant and 1 recessive. And for the table, this is why I'm bringing this up, you need to write both of those, right? So if you have a dominant trait, you need to write both of those in the table. If you have a recessive trait, again, I'll just use eye color. If you have blue eyes, for example, that would be, you would be homozygous recessive. So that would be two little letters. Any questions about this slide? Again, this one's very important, very practical because of what we're going to do, what we're going to be doing in lab. All right, if there's no questions, we'll move to the last one, number 4. A sperm or egg carries only 1 allele for each inherited character because the 2 alleles for a character segregate during the production of gametes. It's a really long-winded way of saying what you already know. You already know about meiosis, right? So in meiosis, if we're talking about humans, right, we have 23 pairs of chromosomes. They all get lined up in metaphase, but then they get separated in anaphase, right? So you start off, that cell started off with 2 different versions, right? The version you got from your mom, the version you got from your dad. But then they separate, right? We separate them in anaphase. And this is known as the law of segregation. You need to know the law of segregation. I should draw a line here. So the law of segregation is up here. Right, that is the law of segregation. Now, when the sperm and an egg unite, Each one of them contributes its alleles, and that restores the paired condition in the offspring. Again, everything you already knew from the last chapter when you learned about mitosis. Now we're just looking at it from a different context, right? So now instead of looking at the chromosomes themselves, we're looking at the actual genes on the chromosomes. And this next slide I'm going to show you is a great way to end the lecture, is the visualization of what I'm talking about, right? So we have a purebred purple flower crossed with a purebred white flower. Actually, you know what? That might be a good thing to start with when we come back. Yeah, so we'll start with this when we come back. The last word for attendance, again, as a reminder, to ask me a question about the material that I covered. Of course, you can ask me any questions you want, but for the sake of the attendance word, you need to ask me at least one question about the material that I covered. Even if you understood everything, maybe come up with a question that I could use on an exam, if you want to look at it that way. But that's it. It's 8:50. You guys have a good week. I will be online for attendance. For office hours. I'm here if you need my help. Happy to help you. Oh man, I didn't even do attendance. Hold on. Really quickly, what do we have here? Kate Stolt is here. Mankins, right?
Yes, sir.
Thompson? What's your last name?
Hughes.
Hughes, yeah, sorry. All right, you guys are good.